comes from material science background as an undergrad, where he did his work, where he did his undergrad at IIT BHU, and then went to Caltech for his PhD. And then he did his postdoc with two of the legends in the chemistry and biophysics, computational chemistry and theoretical field, Professor Michele Paranillo at ETH Zurich and Bruce Byrne at Columbia. And in 2017, Pratyush started his lab at University of Maryland. Within a very short period of 5 to 6 years, he was promoted to a full professor and now leads a very vibrant and across the globe well-known team, um, and he's the leader of the Center for Therapeutics Discovery at the recently established Institute for Health Computing. And I know Pratyush from his work, many work in metadynamics and some very interesting papers that he has published on projection techniques and various other, and definitely not know all the work that he has been recently doing, which is a great opportunity for me to catch up with. And so, Pratyush, without much ado, I know the list can go on and on. I'll leave the floor to you, and I'm sure a lot of people would like to hear your talk, and particularly the kind of topic that you have chosen is probably sort of reassuring to a lot of us who have not yet caught up onto the machine learning and are asking the question like, are we still relevant? So thanks for choosing such a topic and doing the research on such a topic. And the floor is all yours, Pratyush. Thanks.
Okay, can I share my screen?
Yes, let me give you access for that. Yeah, please go ahead.
Okay, great. So I will get started, and let's do this. You can see my screen now, right?
Yes, we can.
Okay, great. So just 1 second. Let me remove all this. Right, video. Okay, great. So I'll get started. So first of all, many thanks to Anand and to Jagannath for giving me the opportunity to speak at this very famous session. I have been watching the videos and following all the people who have spoken here for a very long time. I guess I was waiting for you to switch from Microsoft Teams to Zoom before I come and speak. And yeah, I mean, Anand and Jagannath and I, in a strange way, go way back, way long back in the day. You know, Anand and I, in fact, have the same middle school, believe it or not, a few years apart, but we were in the same middle school, while Jagannath and I overlapped in Bruce Byrne's group in New York City. And I think Jagannath and I, we have at least 3 papers together, all 3 of which were a lot of fun to work on. So yeah, so I'm very happy to be here, and I will tell you some of the things my group has been doing. And it's a perspective from the it's a perspective of what does it mean with all this generative AI happening recently, and what does it mean for doing careful long simulations? Yes, we know molecular dynamics simulations sometimes do not converge. What does that mean? Should we still be doing it? Should we be developing new methods? Where are we going with all of this? I will talk about all of this from the light of what my group has been doing, which is of course a very biased perspective, but I will spend a good amount of time raising some general questions, which I think purely AI tools are incapable of answering in their current form. And I would like to argue that they will never be able to answer these questions, because simply speaking, there will never be enough training data to answer the type of questions we need to answer. So as Anand mentioned, I'm in the University of Maryland, College Park. It's College Park, for those of you who don't know it, is in southern Maryland. So the most famous city in Maryland is Baltimore, but it's actually quite relatively far from Baltimore. It's closer to Washington, D.C. In fact, if I go for a long run, I can run from my house to White House, which is so it's really close. And here I'm in 3 departments. I'm physically located in this institute known as IPST. This is an institute which has had a long history of statistical mechanics, back from Zwanzig and Michael Fisher to Chris Jarzynski and John Weeks. So it's a very exciting and reputed place for young people to come and learn statistical mechanics. So I've been very lucky to be there. So, and my group is quite interdisciplinary. I have students from biophysics, chemical physics, and even applied mathematician who is now at Flatiron. And I'd like to claim that my group is the first group to have a postdoc. That his name is Pakoda. So you can see I miss Indian food a lot, especially my wife misses it, even though she's not Indian. So we named him Pakoda. So, and these are the financial sources that support my group, and this is the conflict of interest. So let's get started. So big picture, my group is interested in three classes of problems. One is protein small molecules based drug discovery. This is a kinase. And as I mentioned, it's Jagannath who really got me the kinase bug. I think my second or third paper on kinase is second and third paper with Jagannath. And back then itself, this was 2015, we started thinking about how can kinases take different conformations? How can we exploit this conformational flexibility to understand why certain drugs bind, why they do not bind? How can mutations come and affect this conformational flexibility? And really a dynamics-based perspective, which is not just a single structure paradigm, but how do proteins change different shapes? How do drug molecules take time to get out of these proteins? And how does this depend on the shape the protein is in? Calculating these things. Of late, in the last 1 or 2 years, I have become extremely interested in RNA because they are just so fascinating. You know, when I was growing up, as Anand mentioned, I was trained as a material scientist. In fact, my undergrad was in metallurgical engineering, so even more old school. I never knew there's nature can have things such as a triple helix. I thought DNA takes a double helix, and that's kind of cool. But RNA can often adopt a triple helix, and this is just one part of the story. They can adopt really complex structures, and they have evolved to be this way that they can take really, really different forms, not stay there for a very long time. And there is a whole area here where we can think about designing drugs that target RNA, and we'll talk a little bit about that. And the third part of my group is interested in problems such as crystal nucleation, especially what happens in crystal nucleation in the presence of in nano-confined environments. So today I will primarily talk about proteins. So, and in all of this, we are guided by structure and dynamics, like I said. So there are three ways to approach biophysics, right? You can think about a purely sequence-driven approach. Some people do that. Or you can think, well, sequence is not sufficient. I have to think about structure also. Or at a third level, you can say, structure is also not sufficient. I have to think about dynamics. Does it stop there? No. The fourth one is in the environment, right? If you even account for sequence, structure, or dynamics, there is still the missing element, which is environment. To some extent, you can account for environment by treating dynamics in a non-Markovian manner. So I would say that if you're looking at structure and dynamics, you can really cover biophysical systems in very robust and different environments. So, and there has been huge progress in all of this with AI. So the less than 4 years ago, AlphaFold came out. It's not been a long time. And DeepMind, when it came out, called it as a solution to a 50-year-old grand challenge in biology. Soon thereafter, there was a paper by Riju Das's group at Stanford for RNA structure ranking more than prediction, which was the cover of Science. And this was titled as Winning Solution for RNA Structure Prediction. So this was 4 years ago. And in the last 1 year, or perhaps in the last 6 months, there has been a complete explosion. And it's become impossible to keep up with the literature. Even for someone like me with a group who is fully interested in this problem of how do we combine AI with molecular dynamics to get dynamics and things like that, it's a complete explosion of problems. I want to highlight a few of these things that I think are interesting, worth mentioning. There are many more. One is, of course, AlphaFold 3, which came out less than a month ago. I think it was end of May when it came out. So less than 2 or 3 weeks ago. And this was, I have never seen so much publicity for a science paper. New York Times, Washington Post, everywhere. And you can see the titles speak for themselves. A few months before that, Frank Noyes' group, and really not Frank's group so much, but his team at Microsoft came up with this method known as distributed graph armor in nature machine intelligence, where the idea was you use diffusion models and graph neural networks, and you can predict equilibrium distributions of molecular systems. So see, we are starting to change perspective from a single structure prediction to equilibrium distributions. Slightly before Frank's group, also in nature machine intelligence, Anima Anand Kumar and Tom Miller, who is now leading a company known as Iambic Therapeutics, they published a method known as neural plexer, which is also approach to get ensembles. And less than 2-3 months ago, MIT, Tommy Jaquela's group published a method known as AlphaFlow, which is again protein ensemble. So I'm highlighting all these things to give you a couple of perspectives. Number one, the field is insanely active. It's becoming very hard to think, how can I even be relevant in this field where there is a breakthrough paper every week? What do? I can't even read the paper at that pace. How do I'm going to compete with this? Secondly, notice I'm not showing you the affiliation list here, but this paper is from Microsoft. This paper is from Iambic Therapeutics. This paper is from Google. We are no longer Anand and Jagannath and Pratyush hanging out at a reaction coordinate conference and thinking how to solve problems by getting a good committer. The landscape has changed. These are big companies with enormously deep pocket who are trying to solve this problem of how to get conformational ensembles and dynamics. So it almost makes you feel sometimes hopeless. Like, what do I do? How do I compete with this? And it makes you feel as if molecular dynamics, age-old molecular dynamics of running a simulation, figuring out your coordinates, doing your sampling, reweighting it, plotting potentials of mean force, maybe this is all no longer needed. Is that really true? Is molecular dynamics dead? Not quite. So let me show you what are some of the things which all these methods, and I'm not really going to talk about my concerns with distributed graph armor or neural plexer. I think they're fantastic methods. If there is time in Q&A, I can tell you why they are very good step forward, but barely tip of the iceberg. But I will show you what are the issues with AlphaFold 2. I cannot really talk about issues with AlphaFold 3 because the code is not out there for us to play. So when we talk about biophysics, it's not just about predicting one structure. It's really trying to understand the ensemble of structures. So for proteins, for ordered proteins, at least, not for IDPs, and Anand can talk more about that. For well-behaved protein, we expect there is a funnel landscape. There is a most stable native structure that the protein takes. There is a bunch of non-native structures. And as you start relaxing your energy of the system, you can get entropically more non-native structures, right? So there are many of them. For RNA, the picture is a bit more complicated. There is no such one native, and maybe IDPs fall in this category. You might have one native structure, but there are many other competing structures which are not that much higher in energy than these structures. And when I say energy, I'm being very casual here. My favorite subject is thermodynamics. That's what I teach. I'm not really getting into, am I talking about free energy or potential energy? I'm being very, very casual in this. Okay, some energy measure. So RNA, you have a very glassy landscape where it's not clear if there is one native structure or what's really going on. Basically, there is no one. It does not make sense to talk about just one structure. There's an ensemble of structures. The barriers of interconversion between any two structures is not as high. Quite possibly, it's on the order of a few KT, not more than 10 KT possibly. But it's enough that if you get trapped, you are probably going to get trapped. It's a very rough landscape. And crystal nucleation, which is the third part of my group, is like the combination of protein folding and RNA. It combines the worst of everything. And we won't be talking about that today. So to really say that we have used AI or any method to understand biophysics, we need to understand this ensemble of structures, and we need to understand the dynamics on this ensemble of structures, because even if you project an ensemble using some free energy, you don't know what are the missing coordinates. You do not know whether this geometry is Euclidean. If this geometry is not Euclidean, then 2 points that are close to each other are not really close to each other, right? You have to be careful about it. So there is a lot that remains. And if we really want to claim that AI is predicting biology, we need to understand the relative probabilities of these conformations in the right order of magnitude, at least. And secondly, we need to understand what is the time scale of interconversion between these. So, and these things have huge repercussions. It's not just a cartoon that I'm drawing for you. Well, this is another cartoon, but this is a bit more practical. This is from Ruth Nusinov's fantastic review in Current Opinion in Structure of Biology last year, where Ruth says, if you have some protein, some generic protein, and let's say you have a wild type, and this is some free energy where the inactive is more stable than active, you will end up in a healthy cell phenotype. If the active is more stable than inactive, you can end up, the cells might end up with cancer. If the active is again more stable than inactive, but not by that much of a margin, it might end up in neurodegenerative diseases. And so again, you can see it's not just about predicting the one most stable structure, it's about predicting what is the relative probabilities of these structures. So how does AlphaFold do this in things like this? So let's start with protein kinases. And this is the this is the system that I will be talking more about today. So for those of you, most people in the audience possibly know protein kinases. So I apologize if it's obvious to you, but I will repeat it anyway. So protein kinases are around 5% of the human genome, something like that. They have a whole term known as kinome. They are the number 2 drug target out there. They are involved in converting ATP to ADP. So there is an ATP pocket over here. And for the ATP molecule to go and bind over here, this activation loop, which is the disordered thing that you are seeing over here, has to be in this extended conformation out here. And this little motif of 3 residue, DFG motif, has to be facing in conformation. It is not facing the solvent, it is facing inside. So this is the fully active kinase conformation. In nature, most kinases try to take such an active conformation. That's how they function. So the question is, do all kinases do this? Do all kinases predominantly try to take active conformation? The answer is not entirely clear, but very strong evidence suggests that it is not the case. Certain kinases, such as DDR1 kinases, and I will explain this pie chart to you in just a moment, but stay with me for a bit. So I'm talking about 3 kinases: DDR1, Abel kinase, and Src kinase. So for DDR1 kinase, through experiments and through classical force fields with extra long simulations run on Folding at Home by John Codera and others, it has been documented that really the kinase can take different conformations. The DFG loop can stay in, out, or inter. It's not that only one state predominates. For Abel and Src kinase, one state predominates. So the experiment we did was to take AlphaFold, and you can, so AlphaFold is a sequence alignment method, okay? So what you're doing is to look at length of sequences and compare it with other proteins out there. And what you can do is to subsample this. So in a way, in a crude way, you're making AlphaFold weaker. So when you make AlphaFold weaker, you are forcing it to generate other structures. You can generate conformational diversity. And through this, you can run it to generate thousands of structures. Notice you cannot do it on AlphaFold 3 because there is a web server. But if you just run the 10 structures that they allow you on AlphaFold 3, the results are not entirely different. It seems that it has not solved this problem. So for AlphaFold 2, if you run 1,000 simulations for each kinase for a very subsampled MSA, if you don't subsample this much, if you stay at 64 or something like that, then you don't even get this diversity. You basically get no diversity. To AlphaFold, all kinases are similar. It is like someone in the audience looking at Jagannath, Anand, and me and saying, this is an Indian person. That's it. It cannot really distinguish that there are differences between Jagannath, Anand, and me, right? It cannot go to a finer level and see that maybe Jagannath has much more hair than Pratyush does, for the things like that. So it's not able to do that. It's just learned what a kinase kind of looks like, and it produces a picture where even when you get diversity, there is no sign of Boltzmann diversity over here. For RNA, the problem is even harder. And this is the only slide on RNA I have. I'm not going to talk after this, but I want to show you how hard the problem is. Also, my group is working on it, so we are very excited about it. So for RNA, the number of structures in the PDB, I need to update the slide, is close to 200. It's now crossed 200,000 for proteins. For RNA, the structure is still less than 2,000. So the number of training structures that you can train on is very, very few. So last CASP competition, so for some of you might know that AlphaFold famously won the previous CASP competition. That was CASP 15. So in CASP 15, when Riju Das and Eric Westhoff and others wrote this survey on how did the different methods for RNA structure prediction do, the conclusion was the predictions for deep learning approaches were significantly worse than the other bioinformatics and other type approaches. So CASP 16 is currently underway. My group is also taking part in it, so we'll see how we do. AlphaFold 3 came out right when CASP 16 started. They provided their results on the CASP 15 RNA, and at least on those, for some of them, it heavily underperforms, but even for which they did not even provide results. But the ones they do provide results on, it's not a breakthrough. It's not a breakthrough. It does at best, it does not, it's maybe in the top 40% or so of the methods that were used in CASP 15. So there is a lot to be done here. And finally, the third challenge that I exist. So just to summarize so far what I have shown, I told you that with AI approaches, you cannot get protein conformational diversity. You can, if you get some diversity, it's not Boltzmann diversity. For RNA, the problem is hopeless almost, because the number of structures for training data set that are out there are just so few, and they do not reflect the number of RNAs you actually have in the human body. If you count the number of mRNA, they are way more than the number of proteins. So there is a misbalance here. And number 3, this is a challenge that my group is interested in, and which maybe I will have time to talk about later, but I just wanted to introduce it, which is this idea that if we are using generative AI, specifically language model, to predict things like sentences, these days, anytime I write an email or anytime you write an email, it's extremely unlikely that you have not made use of text prediction. In fact, this has been going on for the last 10 years almost. Google's autofill has become more and more powerful. And if you write a sentence, it will tell you what to write next. And as you keep using these tools more and more, it becomes more and more powerful, making things very easy. And now transformer-based models have taken it to another level. So the question I'm asking here, if you look at a time series coming from a molecular dynamics, let's say this is a protein, and you study the movements. In this case, this protein is AIB9. It's a synthetic peptide. And let's say it's moving from a left-handed helix to a right-handed helix. And you have a time series where, let's say through this variable chi, which captures whether it is left-handed or right-handed, you can try to run text prediction on this chi, right? Just like you use language models to predict sentences that you have written. A molecular dynamics trajectory in some abstract sense is not really all that different from a sentence. And if in these two papers, we showed that you can use language models, and this was somewhat of a primitive language model that we used, but I think these results apply for transformers also. They are powerful, but they are maybe too powerful. If you show it a time series where this peptide, which is supposed to be symmetric, due to undersampling, if your peptide had a site asymmetry where the left-handed helix was less stable than the right-handed helix, and now you train your text prediction using LSTMs or some other model on this, it will memorize this error. And you cannot blame it for it. It saw it in the time series that there is a slight asymmetry between this and this state. So it just reproduces it faithfully. So, and this is a problem. So if I have time later, I will show you how in this paper, we started thinking of how to come up with approaches which can combine intuition and human knowledge in language prediction models to make the best of both worlds. But so these were the three classes of problem I showed you in the introduction part of my talk. We have proteins, we have RNA, and we have this abstract question of how to use language prediction tools to really predict non-Markovian dynamics arising in biophysical simulations or experiments. And for all of these, my group developed different methods. It will take me a lot of time if I had to walk you through all these methods and show all these things they can do. The methods are work in progress. In no way we have solved the field. And these are very exciting times. All the codes are out there on GitHub and on our website, especially for the students in the group. If nothing would make me happier if you take one of our methods and improve it and come up with a more powerful version. That will be fantastic if you can do that. So please take a look at this if you're interested. Today, I will primarily talk about this first class of method, which is this information bottleneck approach, which we call the RAVE approach, which allows us to speed up sampling in a systematic manner. I will talk about this because this is where we have been able to scale up the codes in a way that now I think other groups can also start using it systematically. So the idea behind RAVE is the problem that we are trying to solve here is to speed up molecular dynamics. Why do we want to speed up molecular dynamics? Because, well, it's very, very slow. And this is why generative AI tools are very appealing. All the tools that I showed you earlier on, Neural Plexer and Graph-Farmer or AlphaFlow, if you have a good trained MD model, then generating new trajectories is much faster than running molecular dynamics with its slow time step of 2 femtosecond and trying to get to very slow processes. So molecular dynamics is slow. It's really, really slow. You have a 2-3 femtosecond time step if you're doing classical dynamics. And with supercomputers, maybe you can get up to a microsecond. With GPUs, with Anton, you can start getting to millisecond. But a lot of problems, such as protein conformation change, crystal nucleation, or the dissociation of a drug are really, really slow. Maybe if you if you use Anton, so a lot of D-Shaw papers end up being papers where they have run simulations for weeks and they get 1 or 2 events. These are the fastest events, especially in the drug unbinding category. All you have done is to sample the fastest event over here. Is that really representative of reality? I don't know. It's just one sample from a whole statistics. If you really want to talk about the process, you need to capture not just the fastest process, which your brute force computing gets you to. You really want to capture a spectrum of processes with their whole timescales. And that is really, really hard. So in order to do that, people have been developing a range of enhanced sampling methods. So in molecular dynamics, the problem is that you're trapped in whichever state you started in. So notice I'm talking here about some projection. What do I mean by that? If you are interested in protein conformation change, for example, This could be radius of gyration. This could be the number of hydrogen bonds, the number of native contacts, something like that, right? So you have some projection, not necessarily the reaction coordinate. This projection is not necessarily capturing all the slow things that happen in the system. It is capturing some slow things that happen in the system. And when you project your molecular dynamics along such a projection, you will see that it is trapped in some basin wherever you started from, and it will be a rare event that takes us across to this other basin. So there are these enhanced sampling methods or enhanced molecular dynamics methods. Here I'm showing you metadynamics, where you start filling up the energy landscape. And notice now I have switched from some projection to reaction coordinate. What do I mean by this? A reaction coordinate is a concept that we all see in our chemistry textbooks in high school, right? It quantifies the progress along a reaction pathway. How much did a reaction happen? It's really hard to pinpoint beforehand for what is a reaction coordinate for conformational change. People still argue over it because it's not clear. In theoretical chemistry, there is a perfect reaction coordinate, which is known as the committer. It just so happens that it is also very hard to calculate. In order to calculate the committer exactly, you need to have sampled the full energy landscape to some more or less. So that makes it a useless approach if you really want to speed up problems. And metadynamics, what I'm showing you over here, is just one enhanced sampling method. There is a whole zoo of enhanced sampling methods. So by the way, if you're watching this talk and if you're in the audience, and if you can successfully name, without looking it up on the internet, every single acronym that I have over here, then next time I see you, I can buy you a cup of coffee or tea as a prize, because it's a zoo of methods. I'm an editor on this journal known as JCTC, and every week we get a new enhanced sampling method, possibly more than every week. And one of the first things all authors seem to claim in their next method is, oh, we do not need a reaction coordinate. Our method is going to work without a reaction coordinate. And generally, it looks like that for very small systems. As people start to use it on new systems, on complex systems, invariably, you need to know what is this low degree of freedom. And notice some people might argue here that why have I put up replica exchange with solute tempering in this category? It's because even this solute tempering, you're kind of assuming which is the part of the system, which is really the slow part of the system. So if you just do replica exchange, and that's a method that is possibly the only method which does not need a reaction coordinate, but you do not get this much speed up. So for really disordered systems, the only hope is to do replica exchange. And that's also something my group is working on how to speed that up, but we will not talk about it. Today, we will mostly tell you, I will talk about the method where we can learn an approximate reaction coordinate using machine learning based approaches, and then supplement AlphaFold with such a reaction coordinate to speed up the process of conformational sampling. That's what I'm going to show you as the example. So why is it so hard to find a reaction coordinate? Shouldn't it be that if you have a coordinate that projects out one state from another state, if you have a coordinate that can clearly cluster out different states from each other, isn't that sufficient? Generally, no, because reduced representations are easy to make. You have such a reduced representation over here, where for some abstract system, you have a free energy along some coordinate R1, and you are seeing 6 states. And let's say I tell you that this system actually has only 6 states. Does this mean R1 is now a perfect reaction coordinate? And I would have loved this if I was giving this talk live, because normally I like to poll the audience for this, and you would have so the question I ask the audience is this a transition state or not? So you can answer it yourself. This is like an open book exam that you have to grade yourself, and just take a moment to think, is this a transition state or not? Okay, and I'm not asking you to answer verbally. I'll just have some water. So normally when I ask this in talks, roughly half the people raise their hand. Yeah, that looks like a transition state. And it turns out it is not a transition state, because this projection is coming from a higher dimensional projection where I have removed the R2 degree of freedom. And the R2 degree of freedom, if you look at that one, then you realize that the states are actually connected this way. One is not connected to 5, which is true over here. One is not directly connected to 5, but 2 is not connected to 4 either. And if you look at this projection, the transition state that you're getting between 4 and 2 is spurious. It's just a projection-led mistake. So this reduced representation can be thermodynamically correct. There is nothing wrong in the thermodynamics of this projection. You can marginalize, if you can write down an analytic, and you can do this exercise, right? You can just write down 6 delta functions, you can do the averaging, and you can come up with something like this. And the relative thermodynamics is perfect. There is nothing wrong in it. In fact, the thermodynamics of this point is also perfect. There is nothing wrong in it. However, the dynamics is completely misleading. This is not a transition state. You never go from 4 to 2 directly. You always have to go from 4 to 5. So this picture shows us that even in the simplest systems, it's hard to come up with projections that are mechanistically sensible. And now if you try to do umbrella sampling on such a projection, most likely your results will be wrong. You won't even realize it because umbrella sampling can be very, very dangerous. Jagannath has this beautiful paper that he wrote with Richard Friesner and Bruce Byrne, where Jagannath did in 2016, Jagannath did this umbrella sampling simulation on the dissociation of a drug from a protein. And he did umbrella sampling along the distance coordinate, and he got a very nice free energy like this. And then Jagannath added a water coordinate, and he showed that the landscape completely changes. So we have to be very careful. In methods like umbrella sampling, you can just run VAM, and if your coordinate is missing out a slow degree of freedom, you won't even realize that what you have is incorrect. Metadynamics gets a lot of criticism because people's trajectories do not converge. But to me, that is a reason why I'm still continuing to use metadynamics, because it is self-diagnostic in a sense. In metadynamics, if you're missing this R2 degree of freedom, you will see that you just get trapped somewhere, unlike umbrella sampling. So now, even in such a simple system, it's so hard to come up with a projection that is correct, right? And as I mentioned, this is due to this projection error. So now we have this whole zoo of machine learning methods like TSNE and UMAP, where we can project anything out. So in this case, I'm showing you 2 classes of projections, one from TSNE, one from UMAP. And the underlying object in this case is a mammoth. And just by looking at the projection, there is no way you could have told this is a mammoth. So you can do something on this, but it won't really tell you much about the mammoth. So it reminds me of that story about, like 6 blind people poking an elephant, something like that. So, and now TSNE, UMAP are just 2 methods. There are so many more methods. And all of these methods are very good in clustering. You can get nice clusters. Sometimes you can get very well separated clusters where protein in one conformation is separate from protein in another conformation, but there is no guarantee that you are not falling into this trap that I showed you over here, that I, by the way, shamelessly borrowed from Gerhard-Stock's paper. So it's hard to come up with projections that are also kinetically meaningful. I'm not saying that all these projections are wrong. I'm just saying just because you are getting good clusters does not mean these clusters are mechanistically meaningful. Just because 2 clusters are separated from each other, there is no guarantee that the clusters in the high dimensional space are also actually separated from each other. So you have to be careful. So the way my group tackled this problem was to use an approach. And I'm not saying this is the only way to do this. There are other approaches also. I will show you why we like this approach a lot. This is known as information bottleneck. So what is information bottleneck? It really goes back to a beautiful paper by Shannon of Shannon information theory, the father of modern information sciences and electrical engineering. And Shannon's rate destruction theory in 40s, which was then popularized by Bill Bialek at Princeton in the context of information bottleneck. So I'm using the Bialek language over here. So what Bialek says, if you're trying to come up with a model of a system, so what do I mean by model of a system? Let's say you have some high dimensional input and you are trying to make a prediction. You want to come up with a model which, given the input, allows you to make a prediction. So let's take an example. The input could be the temperature of all cities in India today. So let's say the input is Bangalore, Bombay, my hometown Patna, Delhi, Kanpur, Lucknow, everywhere. You have the temperature of all cities today. And you want to come up with a model chi that tells you what is the temperature of Bangalore tomorrow, okay? Or the input could be, again, the temperature of all cities today, and the output could be whether it is going to rain tomorrow. So the first output was a vector, right? It's a real valued number, which not a vector, it's a real valued scalar. You're talking about a temperature, or the output could just be binary, whether it's 0 or 1. And it doesn't even have to time in it. The output could be, the input could be just photos of animals, and the output could be whether a photo is a photo of a dog or a cat, right? So any general model. Bialek says such a model should have 2 properties. Number one, it should be as predictive as possible. If a model is not doing a good job of prediction, then what's the point of that model? I mean, in India, we just had the elections, and I don't want to talk politics, but there were a lot of models which made a lot of predictions, right? In the end, you like a model whose prediction was correct. Otherwise, that model is not kind of good. At the same time, the model should also be as compressive as possible. What do I mean by that? So let's go to our example of temperatures in all cities and trying to predict the temperatures in all cities tomorrow. Now, let's say to this input, I add, let's say I had the temperature of 10 cities today, and to this I add 10 other information, which is white noise. It's just a random number. So my model should be able to tell that out of this 20-dimensional input, the 10 degrees of freedom that you just added are noise. I don't care for them. It should be able to compress the input and remove the signal from the noise. So you get this double funnel structure where you're compressing the input as much as possible, and you're trying to predict the output as well as possible. In molecular dynamics, especially in this paper, which is the titled State Predictive Information Bottleneck with my student Dedi Wang, we started with Rave a few years ago, but this is really the paper where we got the theory right. We say that for a molecular dynamics system, if the input is past high dimensional coordinates, in principle, this could be the all atom coordinates. Generally, we work with features. So for a protein folding problem, we look at contacts between different protein heavy atoms. For the protein ligand dissociation problem, we will look at contacts between protein and ligand. The reason we look at such features as opposed to coordinates is because coordinates, you have to worry about equivariance and things like that, which makes things messy. If you're looking at such features, you lose some information, but it takes care of equivariance and things like that. So what we show in this paper is that if the input is the high dimensional coordinates of the system at a certain time, and the output is the future state of the system. What do I mean by state of the system? Well, the number of states a system can take depends on how far into the future you are looking. If, let's say, I'm trying to describe myself, I am the system, and I'm to describe what is the state I'm going to take 100 years from now. Probably the only thing you would care for is whether I'm dead or alive, right? So you are thinking about two states, dead or alive. But if you're thinking about what is the state I'm going to take 1 hour from now, there is a whole different range of states that you might be interested in. So the number of states of a system depends on how far into the future you're looking. And I'm going to give you an example to show what I mean with it. And what we show is that if you write down a mathematical function in a way like this, where you have a bottleneck variable, which is compressing the input as much as possible, while still trying to predict the future state of the system as much as possible, and you minimize the mutual information between the input and the bottleneck. So this is mutual information, which is kind of like the covariance. At the same time, you're maximizing the mutual information between the bottleneck and the output. There is a control parameter here, beta. If you minimize this objective function, you recover the committer. You approximately recover the committer, and you can get this to work with very much limited data as opposed to methods like likelihood maximization and things like that. So it's a robust and fast approach, and the bottleneck is expressed now as a deep neural network based on an input trajectory. And some of you are probably familiar with autoencoders. So this is a generalization of an autoencoder. In an autoencoder, the output is the same as the input. You have a high dimensional input. You're trying to reproduce the same thing. And there is no time. So this is a generalization of a time-lagged autoencoder where we are not trying to encode the system directly. We are trying to encode the system so that it can predict the future state of the system. So I'll show you a very, very simple system, which will hopefully give you some intuition. And before I do that, I have to mention one more difference from autoencoders. In training of an autoencoder, you have to assume a prior for this bottleneck. Normally, people in autoencoders, you assume a single Gaussian prior. Here, we assume what is known as a VAMP prior, not to be confused with FAMP noise, VAMP net. This is VAMP prior, also from Max Willing, where the idea is you don't know the number of states a system will have, right? So let's say you have a 4-state system, such as this one, right? A priori, if you have not sampled this, you don't know how many states are there. So let's say you assume this system has 10 states or 100 states or 200 states. You assume a very high number of states when you set up the algorithm. And you then predict the population in different states. More specifically, you predict the transition probability of moving between different states. And I'm going through the math just a little bit quickly, but that is the main objective here. We predict the transition probability of going between different states. And we find that some states disappear. There is no population left in them. So the method tells you through iteration as to how many states does the system has, which depends on the time lag, on the time you're looking into the future. So let's go to this 4-state system. So here you have 4 barriers. You have a very high barrier, you have a second highest barrier, and you have third highest barrier. So if your time scale is actually, we can look at this one. If you're looking 0.5 time units into the future, and this is a model system, so what exactly 0.5 means, it's like a Leonard-Jones interaction, or not Leonard-Jones, it's just an analytical potential. So 0.5 here is something where if you start with population in 10 states and you run the algorithm, you will find that the probability of 6 of these states disappear, you are left with only 4 states. So the method already automatically told you that for 0.5, if you're looking 0.5 time units into the future, all 4 barriers matter, and it basically recovers that you're metastable inside this state. If you're looking 2 time steps into the future, now this barrier becomes irrelevant, right? Because you are equilibrating at a faster time scale relative to this barrier. So that one disappears. Now you're left with 3 states. If you're looking 10 units into the future, you're left with only 2 states. If you're looking 1,000,000 time steps into the future, even this state will disappear and you will be just left with the most dominant stationary state of the system. So what does it mean for practical problems? For, let's say you have drug unbinding. For drug unbinding, you don't know what time step to, how far into the future you should take. So for practical problems, what we do, okay, if you have drug unbinding, we know we are interested in at least 2 states. We are interested in the bound state and the unbound state. So we start with a time step. We start with a very, very high time step. We get only 1 state. We reduce the time step further. Now we get 2 states. So I have a coordinate which is able to demarcate the bound state, number 1, in this case, from the unbound state, number 8. I'm just using this as an example. Now we know that drug unbinding is not really a 2-state problem. Maybe I need to worry about the metastable states that might be relevant in the pathway. So then I reduce the time step further. Normally for practical, and this is a bit heuristic, we stop at a time step where we are getting 3 or 4 states as the stable population. And then we bias it in enhanced sampling and we see what happens. So, and as I mentioned, this thing approximates the true reaction coordinates, the committer, not just for high energy problems, but even for entropic problems. So this is a famous potential by Ron Elber, where the barrier is not surpassing energy. The barrier is finding an entropic bottleneck and going through it, which actually shows up in a lot of biological systems. And here you can compute the actual committer from Markov state models, and you can see how the committer isocommitter surfaces are very sharply separated right at the barrier. And if you just did Tika here, for example, you will not get that because it's a linear method. Tika is just picking up X coordinate. That's all that's happening. So at this point, what have I showed you? I have showed you that the state predictive information bottleneck can approximate the committer. And committer is known to be a good reaction coordinate. A committer captures the relevant slow degrees of freedom. So now we can put it to use. Let's say we have a molecular dynamics trajectory, which is a slow trajectory. Let's say nothing happened. Or let's say it is a biased trajectory where you took your favorite intuitive collective variable, you biased it in umbrella sampling, in metadynamics, in weighted ensemble. Although weighted ensemble does not really give you any speed up, so you might end up using all your computer time if you do that. So generally, especially in the starting phases, it has its own advantages, but it won't give you the exponential speed up that you get in metadynamics or umbrella sampling. So, but let's say you have a biased method and you have a trajectory. You take this trajectory, you minimize this loss function that I showed you over here. In this case, actually, you maximize it. I have a minus sign flip. And you learn this bottleneck. Notice I'm showing you a linear encoder and a nonlinear decoder. When we first started doing this, we thought, well, what will we do with a linear encoder? We must take a nonlinear encoder. But for really complicated problems, we are starting to realize that for sampling, a linear encoder is far preferable than a nonlinear encoder. Why? Because number 1, it's harder to overfit. Number 2, it's easier to interpret. If these are features, and you can see that your linear encoder is giving high weight to some feature that you don't expect to be correct, you know something is wrong. So it's interpretable. So what we do is to take this bottleneck, we performed our sampling. And again, in my group, we are doing all 3 things. We are doing metadynamics, weighted ensemble, umbrella sampling. Each of these methods has its advantages. We use the method depending on the problem that we want to solve. Now we have a superior trajectory with better enhanced sampling. And we take this trajectory and train a new round of reaction coordinate. And we iterate until the procedure converges. What do I mean by procedure converges? Well, the reaction coordinate, the simplest thing you could think of if the reaction coordinate does not change. But that's actually quite hard to quantify. Sometimes the sky is two-dimensional. You have a 2D reaction coordinate. And in that case, you might be just having a rotation. And that does not mean it has not converged. It's just rotating. It's not changing anymore. It's just rotating. So it's hard to say that. So again, in metadynamics and umbrella sampling, this is easy to check because at least in I'll just give you an example of metadynamics. When you're driving the system from point A to point B in metadynamics, you are really, what you're doing, metadynamics is a time-dependent biasing process, right? So in reality, it's not an equilibrium process. It's a non-equilibrium process. And in any metadynamics simulation, you're measuring the work done to take the system from point A to point B. And theoretically, there is no guarantee that you have measured the free energy difference. All you have done is to measure the work done. And second law of thermodynamics tells us that work done will always be more than equal to the free energy difference, right? So as you improve the reaction coordinate, as you start to learn more and more slower degrees of freedom, you do the work in a more equilibrium manner, keeping everything else same. And you can measure the amount of work done on the system through rounds of training of the reaction coordinate. And we stop the training when this work done converges. That's what we do. And we test this on a lot of model systems, like this protein AIB9, where we can fold and unfold it. And this is a problem where the input features are all dihedral. So you have 9 residues. So 9 multiplied by 2 is 18 dihedral. If you take their sine and cosine, that is 36 degrees of freedom. And you are learning a deep neural network. In this case, this was actually a deep encoder. You're learning a deep neural network as a function of these 36 coordinates and biasing this deep neural network. This was work done by my student Shams. The middle one is Shashank Pant, who some of you might know, visited my group. He did this very inspired by a paper by Jagannath, where he folded and unfolded GB1. And ligand permeation, this is not really a real ligand permeation problem. This is the only system we looked at just to show that it can work for a whole range of systems. And now the final example I will show you is AlphaFold. So as I mentioned, by subsampling AlphaFold, you can start generating lots and lots of structures. So this paper was published by Hassan Mekharab and collaborators Diego Del Lamo in eLife, where they showed that for this particular protein, if you stay with the default MSA length, I don't know what the default is actually, but if you stay with a very high MSA length, you get very few structures. As you start reducing the MSA depth, you start to get more and more structures. So when they published it, actually a very talented PhD student in my group, Akash Nathan Aranganathan, he discovered this a few months before them in a rotation project. And I thought, well, okay, that's fine. Why do we care about that? Because this is, you have just weakened AlphaFold to reduce and generate lots and lots of structures. That does not mean these structures are Boltzmann ranked structures. They are just possible hypotheses. So we did not publish it. In retrospect, we should have published it. This paper is very, very well cited now, but that's okay. We were interested in Boltzmann sampling because what you have done now is to generate lots and lots of structures. But let's say you're interested in drug discovery. You are interested in taking one of these non-native conformations and doing docking on top of it. Which one will you take? Will you take this one? Will you take this one? Will you take this one? It's not clear. And docking is still fast. How about free energy perturbation calculations, FEP? You cannot run them on all of these structures. They are just too many. So you need to have a way of Boltzmann ranking these structures so that you can pick up, oh, this is a good representative structure. This is a good representative structure. And from all these diverse structures, I should focus on this 2 or 3. And that is the problem we would like to solve here. So to do this, we treat AlphaFold as a hypothesis generator. What do I mean by that? I'm sure 90% of you, if not more, have used ChatGPT. It's useful, right? Because when you are stuck, it can generate hypotheses for you. Sometimes when I go to a party and I want to make a joke, I don't know what joke to make. Sometimes I just go to ChatGPT and like, tell me a joke about blah, blah, blah. It generates 10 jokes. I'm like, none of these jokes are funny. Tell me 10 more jokes. And every once in a while, one of those 20 jokes will be very funny. And then I can become very popular at the party. Just giving you an example. Now, so it's a hypothesis generator. When you're trying to write a code, for example, you're stuck in something, you can go and ask it, tell me 5 different ways to do this. It will tell you 5 different things very, very quickly. are you really going to bet your life on the answers generated by ChatGPT? Probably not. So the question is, why are you ready to bet your life on structures generated from AlphaFold? If it looks like something that you expect to be true, then maybe you can do that. But then you should be asking yourself this question: if I already knew what it should look like, then why am I using AlphaFold in the first place? So if you really want to do predictive stuff with AlphaFold, I think we are entering a very dangerous regime where we don't know when should we trust it, when should we not trust it. That said, it is a fantastic hypothesis generator. Just like ChatGPT has transformed modern life, similarly, AlphaFold has transformed biophysics. There is no question about it. But for us, it's a hypothesis generator. We can run reduced MSA AlphaFold or AF cluster from Dorothy Kern and Hannah Wymont Steele. And these methods are really not all that different. The answers are very similar. They all generate lots and lots of confirmations. These are our hypotheses. And now we can run RAVE starting from these hypotheses to rank them. What do I mean by that? So we have a sequence. We generate lots and lots of structures from this sequence. And we can learn a trial reaction coordinate, because the problem in the RAVE method so far had been in state predictive information bottleneck is, how do you learn the reaction coordinate if you have never seen the other state at all? And AlphaFold is a very quick way of checking what might be these possible other states, right? So we can generate 1,000 structures from AlphaFold, from reduced MSA or anything, and then cluster it all together to learn a reaction coordinate. We can take this reaction coordinate, we can do our enhanced sampling, metadynamics or umbrella sampling. We can get them as correctly ranked. Then we can even go back and take the and the thing about biology is, just because this metastable state is much lower ranked than these two other, that does not mean this metastable state is irrelevant. What we do is to take representative structures from each of the metastable states, and then we can pass them as a template to AlphaFold and really start introducing more, generating more structures. So we are making, the point I'm trying to make here is that AlphaFold is wonderful, molecular dynamics is wonderful too, and together they are even more wonderful. So with this, for example, for this DDR1 kinase, here I'm showing you just the DFG loop movement, not the activation loop movement. So you are watching it, and this is movie made by my previous postdoc, Bodhivani. She is now a scientist at Prescient Design. So Bodhi is showing here as to how the DFG loop is in the in conformation. And if you just did MD, nothing would happen. But with AlphaFold to RAVE, you can actually get the DFG loop to go to the out conformation. And some of the trajectories in metadynamics, it actually does back and forth. The activation loop is harder to move. And now we can get the right population of the different conformations. If you just did AlphaFold, you get DFG in to be dominant. If you run AlphaFold to RAVE, you get the other states also to be there. And John Corderas and Sonia Hansen in this paper did very long Markov state models to show how does the population of these three states differ with the presence of mutations. And we can also capture those in good agreement with folding at the home simulations. And the final application I want to show you is for docking. This is a paper which is out for review at eLife. I guess that eLife has this nice model where they send it out for review. It means it will get published. So that's a great model. So anyway, we can talk more about that later. So here I'm looking at two kinases, ABL and SARC. For ABL and SARC, let's look at this plot over here. This is very new results. I just took snapshot from the paper. I'm showing you here that if you produce a histogram of structures that you get out of reduced MSA AlphaFold or AF cluster, very few of them have this activation loop folded. So in the in conformation, in the active conformation, the activation loop has to be extended. In the inactive conformation, the activation loop has to be folded. And this is like a 12 angstrom, 14 angstrom movement. It's a huge movement. So if you generate 1,000 structures from AlphaFold, very few of them have activation loop in the right conformation. Furthermore, even though the activation loop moved, this DFG motif over here also has to flip. Now, if you just focus on these, and then you study which of them has the DFG motif which has flipped out, you find that 0. For ABL1, it's 0 of them which do that. And all of we can get confirmations out of AlphaFold papers. Not a single one of them has showed how they can do this for ABL kinase, apart from in this paper where we showed how by combining this with molecular dynamics, we can do this. And this is work done by my postdoc, Xinyu Gu. So what Xinyu did here was to realize that for DDR1 or for SARC kinase, you can actually get some out conformation. So if for one kinase, you can start getting different conformations, then you can use these structures, pass it as a template to generate conformations for other kinases. And I know I'm going a bit too fast here, but the take-home message here is that if you try to dock your ligands, we have ligands which are known to dock to the kinase in the in conformation, and we have ligands which are known to dock to the kinase in the out conformation. On the left, I am showing you ligand docking results to conformations generated from AlphaFold for Abel kinase. If it's a type 1 kinase, you can see that it kind of does okay. For SARC, it does better. But even this is not very good. It's only 4 angstrom. So small side chain placements that are wrong in AlphaFold can really screw up your docking. For type 2 inhibitor, where you have to get the kinase exactly in the out conformation, inactive conformation, it really fails for Abel and for SARC. But now, if we take these possible conformations, we run AlphaFold, we run Rave on top of these, we actually get a PMF, we get a landscape, and now we get thermodynamic ranks of these structures. We just pick the top 2 structures. The top one generally does not work. We find that we have to go to top 2 or 3, which shows force field errors or things like that. I'm not sure what's the reason here, but if you take top 2 or 3 structures, you get perfect docking scores, even for type 2 inhibitors, which is, this is not drug discovery. This is towards drug discovery. This is a retrospective study where we are able to do this. And I don't have really time to talk to you about this, second method where we are adding constraints to language models. But the big idea here is that language models are powerful. Perhaps they are too powerful. They can commit everything to memory, and you can start making deep fake trajectories. So if you have done an MD simulation and your reviewer tells you, oh, your simulation is only one simulation of 1 microsecond, I want you to go run 10 more simulations. You can use language models to fool the reviewer. But that's probably not a good thing to do, right? Because these are too good. These are just deep fakes of trajectories. And while they are powerful, they will reproduce the error that you saw in the training trajectory. So one of the things that we did in the second Nature Communication paper was to use ideas popularized by David Chandler and Juan Garahan back in the day, and even before them, of this idea of tilting trajectories, and where we can correct language models to produce right kinetics by mixing intuition and chemical priors into them. And I don't have time to talk about this. I'm happy to send my slides to Anand so you can go through these and also just look up these papers. So with that, I will conclude. So the take home today is that, well, number one, AI is here. There is no question in denying that AI is a force to reckon with in biophysics, in society, in everything else. However, AI is a hypothesis generator. When you are stuck, when you have no idea what to do, it's a very quick way to come up with ideas. You should not be really trusting your life on it. There should be a form of active learning, or whatever you want to call it, where you take this hypothesis from AI, you put it in a physics-based simulator. You can also put it in experiments. A lot of groups are doing that, like Connor Colley and Alana Speruguzic and many others. And you have to have a feedback where you can quickly rank the hypothesis coming out of generative AI. And I think enhanced MD and so physics-based simulators, I think, are going to be more powerful than experiments in this case, because we can just run through them very quickly, and particularly enhanced molecular dynamics, because it's a very quick way of generating information across timescales. And I think this combination of enhanced molecular dynamics with AI will also help us create force fields, because right now, force fields are fitted to either zero temperature quantum chemistry or to some thermodynamic information. Force fields have not really seen the kinetic barriers, right, when they were fitted. And now if we can use AI and MD to start generating trajectories, which are really long time scale, which can capture non-native conformations, it might be a new era for fitting force fields also. And today I mostly showed you the Rave method. There's a lot of short forms here, so it's easy to get confused. Rave is our overall platform where we learn an information bottleneck and speed it up in enhanced sampling. The particular flavor of information bottleneck that we learn is known as state predictive information bottleneck. And I very briefly flashed this path sampling method, and all our group codes are on GitHub, and I will be happy for any questions.
Yeah, thanks, Pratyush, for a wonderful talk. The floor is open for question now. I already see somebody has typed in the question. Oleg, you want to ask the question, or do you want me to read it out?
You can read it. Let me see. I will stop my screen share and read it out.
Yeah, you can read it.
That will be the best thing. Okay, great. So there is a question from Oleg. Thanks for the amazing talk. How deep is the network to extract the reaction coordinate in the Rave method? Yes, it makes absolute sense. So let's say your data is really poor quality. which is going to be the case for most rare event systems, right? The first round of data that you get out of some short unbiased MD or out of a preliminary biased MD simulations, you are not going to trust this data set, right? So at this point, if you start fitting it with a deep neural network, you are going to capture all the noise in the data set, right? And it's not going to extrapolate well. So in the early stages of training, we start with a linear encoder. We do not go deep at all because it does not overfit. And secondly, linear models extrapolate better than deep neural networks, right? Deep neural networks are very good in fitting the data set you have. They are not going to extrapolate well. So as you keep training, as you start trusting your data, you can start adding depth. Normally, there is no sense to go beyond 2 or 3 layers. That's enough for us. So hopefully that answers the question. How sensitive are these methods to neural network hyperparameters? Oh, there is no deep neural network method that is not sensitive to things like this. So it's always sensitive, but it's particularly sensitive to this beta parameter that I showed you in the information bottleneck framework. So beta is a control parameter which tells you how much do you want to compress and how much do you want to predict. And this is a fantastic research area. There are papers by Max Tegmark at MIT where he studies phase transitions in these models. So there is a whole range of beta parameters where the models are robust. And if you go into another beta parameter, the deep learning itself undergoes a phase transition. Apart from the beta parameter, the other hyperparameters, such as the depth of the neural network, such as whether you have ReLU or sigmoid, it does not make an enormous difference.
Any questions from the audience? You can raise your hand and ask the question. Raghav has a question. So
yeah, while compressing the high dimensional inputs, how do we make sure we don't lose the important inputs in the presence of noise? Well, that is the bottleneck function. So for this, I can share my screen again and show you that is exactly the purpose of the bottleneck. So let's go back. So if you look over here, the objective function that we have is the difference of two mutual information. So this first one is the mutual information between the bottleneck and the input. And the second one is the bottleneck and the output. So like you say, how do we make sure we don't lose the important inputs? The important inputs are the one that are needed to maximize this quantity minus IXY. If you lose the important inputs, you will not be able to get high mutual information between the bottleneck and the output because you will have no predictive power. So it's through this objective function that you do that. So exactly. So for example, if, I think I will just stop at that. So this objective function, it's due to this trade-off of these mutual information that you're able to do that.
there is more question coming in.
Yeah, I see them. I'll keep going. So I'm curious to know that for IDPs, MD simulation is a slightly different ball game. How much do you think applying encoders can help in studying the conformation ensemble of IDPs or the low infinity high specific interactions that they generally take part in? Oh, hopeless. It's not going to work. So my group does. Let's see if I have those backup slides. So my group looks at RNA. And Anand, please correct me if you think I'm wrong, but I would say RNAs are kind of in the same category as IDP.
I also tend to think like that. Yeah, I think it's a good way to think of it.
Yeah, to use language from my home state Bihar or if anywhere, it's like the Chachera Bhai of RNA. It's not too.
Even when G.D. Sponer was giving one of those talks, and he also tends to think that there is an ensemble average that's more closer to IDP than a folded protein structure. So
yeah, exactly. So for RNA, we don't do autoencoders. We do replica exchange and there we have a whole different class of methods where we speed up replica exchange by using something known as diffusion models. And if I post so these are diffusion models where we really draw parallels that the sampling of spin glasses is very similar to the sampling of disordered systems. And that's a whole class of methods. The first one was published in PNAS in 2022. This one is out for review. And you can read that. That might be useful for IDPs. Even there, we are starting to realize that some amount of dimensionality reduction still might help, but it's not an encoder. So, and I can tell you something here. So for example, DALI, which is this method where you can go and put in text, right? Give me a picture of a cat wearing sunglasses, and it uses these beautiful looking pictures, which are very hard to tell whether it's true or not. In fact, on Facebook, on different forums now, a lot of photos are actually predominantly AI generated photos. It's becoming more and more, it's becoming hard to distinguish. Facebook algorithms are unable to rank whether it's AI generated or human generated. So these algorithms, they do not do dimensionality reduction. When they first started, they would work with the full high dimensional space. But DALI actually does do dimensional reduction. DALI does something known as stable diffusion. So even for IDPs, there might be benefit in doing dimensional reduction, but it will be different from autoencoders. And if you really need to speed up IDPs, I think, and people in this audience know way better than me, the approach is replica exchange family of approaches. And how to use AI to speed up replica exchange, I won't do that using autoencoders. I would do that using, well, we do that using diffusion models. So that's a long answer to your short question.
So, Pratyush, following up on that, I was, something caught my attention which you didn't talk about so much is that you said replica exchange with no exchange. There was in brackets something like that. Can you extrapolate on that? Like, so
Yeah.
you
So
said that thing, right? Replica exchange with no X. So in bracket, I thought I
yeah,
saw.
yeah.
So that.
Absolutely. I will just show you the result. So let's look at this example. So Ising model, which is like every StatMech person's favorite model. And we show this in this paper. So let's say you have sampled the Ising model at two temperatures, one in the deeply magnetic phase and one in the deeply non-magnetic phase. And you have generated ensembles of spins at both temperatures. You know that in a 2D Ising model, there is a critical temperature. And if you're like me, you probably remember the value of this critical temperature. It is 2 by natural log of 1 plus square root of 2, which was Onsager's result, for which he did it in his PhD and then later got the Nobel Prize for it. So you know this number, but the question is, if you just show information from these two temperatures and you don't show any information from system sampled between these two temperatures, can you reproduce the whole magnetization versus temperature curve and even the spike in the critical heat capacity and things like that? And with this diffusion model, so this is replica exchange without any exchange. So this is two ensembles of the system where there is no mixing of information, and yet we can generate information. So how does this work? And since you asked Akash, Anand, I will tell you very, very briefly. So what does replica exchange do? Replica exchange is a way of passing information between different replicas, right? You are exchanging information and you're trying to cause information to flow from the hottest temperature to the coldest temperature. As you know, the 1 over square root of n curves, as n becomes higher and higher, this information flow goes to 0 and nothing percolates. So that's why you have to do solute tempering, where you don't exchange information about everything, you exchange information about, or you don't heat up everything, you heat up only parts of the system. But if you do solute tempering and if you have big conformational changes in the system, it's not going to work. This is well documented. So what we do is to take a very different approach. And since Anand, since you asked me this question, this is a pop quiz for you. Hope you're ready. Know the name of this character?
That's from the Getter Fix, is it?
Yeah, there you go. It's Getafix. It's my favorite cartoon character. So think about Getafix. So Getafix has this magic vessel in which he can throw anything, and he seems to like throw whatever. He just sends Obelix and to the jungle to pick up something, and he throws it, and he can generate whatever out of it. So we are going to take a different perspective from replica exchange. We are going to think about replica exchange from a post-processing perspective. So let's say you simulated your system at 4 temperatures and you did not get very good mixing. Is that hopeless? Does this mean you got no information? Actually, there is still information that you got. What you have done now is to sample X, and this X is X. Ideally, X can be all atom coordinates, but again, you run into this equivariance problem. But this is not a reaction coordinate. For an RNA, X tends to be like G vectors popularized by Giovanni Busi, or something like that. It's very high dimensional. It's 100 dimensional, perhaps, something like that. So what you have done is to sample X conditional on different temperatures where you did your simulation. And you throw all of this into Getafix's magic pot, and you ask Getafix to use generative AI to generate samples X at any other temperatures of interest. Not just the free energy at that temperature of interest, but samples, its actual sample generation. And this is what we are able to do using these diffusion models. There is a bit of iteration. So what we do is, even though there is no exchange, Once we generate these samples, we do a new round of molecular dynamics. And this is the tricky part, which is very important. So your system is so large that you're not going to get exchange. But what you can do is to initialize your system in very smart ways, right? So initially you initialized it in some random way. But now GetAfix has given you samples conforming to that temperature of interest. So you take this temperature of interest and you reinitialize your samples on that basis. And then you keep iterating. So you're doing rounds of no exchange replica molecular dynamics with rounds of training a diffusion model. And through this, you can get converged free energy surfaces. For example, I showed you the Ising case, but I can show you, this is an RNA, for example, this is HIV tar, and it's in this paper, you can see. It's kind of similar to an IDP in some sense, and here we can do this process and we can see we are getting the free energy on different temperatures with no exchange within the different replicas. But what we are doing is to iterate between rounds of replica no exchange with rounds of training the diffusion model. So it's not going to be as fast as metadynamics, but we are very excited about this method. And
Yeah,
I think
it's quite exciting. So we are
yeah,
stuck with this 120, we have been running with this solute tempering. So,
yeah,
so there's some replica exchange method that we developed with hybrid tempering.
I
And
remember.
for
Yeah.
larger IDPs, it's a nightmare. For the last 1 year, we have been, maybe we should read your papers more
So
carefully.
you
And
can read it and you can try the codes also out there. We can help you with this. These are,
yeah, I will get back to you.
as you know, Anand, very well, we are not DeepMind, right? So in our academic group, it's hard to come up with code which is immediately usable by everyone else,
I
right?
agree with you.
By our
Yeah.
best. Because if you just do that, then some people will come and be like, well, that's
Yeah.
not theoretical chemistry. You are just doing software development, right? So it's a challenge.
And so the topic barriers are very, very acute with as you increase, like, at the higher replica. So I will probably go through this and try
So
to.
go through this. Our software might not be exactly what you want to use right away, but we will need to help you with it, and we can do that. So my point being that the code exists on our
Yeah,
GitHub,
right,
and
right.
you will. So don't just try it on your own right away. Email us what your thing is, and then we can set up a Zoom, and I can help
I
you.
understand. Yeah, I'll do that. One more question before I let Nilanjana ask. So I
Yeah.
was struck by your comments when you are talking about, you're so this free energy always being less than or equal to the work done, and then eventually you make sure that the work done is converged. It sort of so you have been also been talking about non-Markovian methods, which basically means there's some
Correct.
non-equilibrium kind of things going on. So it kind of is have you like probably you had already thought about it. Is there a connection to the Jardinsky equality and those kind of things in this framework where you are
Sure,
sure that
sure. People have tried to use Chris's relation, and Chris is my colleague, he works on the floor above, to get better estimates from metadynamics and these other non-equilibrium processes. The problem is, even though Jarzynski's relation is one of the most, I don't know why I'm scrolling back.
celebrated sort of equation, but converges
It's
or
the
it,
most celebrated equation,
yeah,
but uh, what it says is that the mean of the e to the power beta delta f is equal to mean of e to the mean of
initial of mean. Yeah,
power delta w, right?
yeah,
It's the mean. The variance is infinite.
yeah.
That's a problem. You cannot sample it accurately. So Crookes relation makes it a bit better
Yeah.
because so
Forward.
think about it. It's a celebrated relation, but you're trying to converge a quantity whose variance is infinite. Good luck with that. That's going to be really, really hard. It's the fattest tail distribution that you can think of. It's not a Poisson statistics.
Mhm.
Crookes relation is useful because now it's telling you, yes, it is Poisson statistics, but drive it in forward direction, drive it in backward direction, see where the tails intersect. So Crookes makes it useful, but that becomes hard to implement it over here. So that's why, while it is the most celebrated relation, it is hard to implement it, but it gives us a guidance to how to do things.
I see. Okay. Okay, I'll let Nilanjana. Sorry to keep you waiting, Nilanjana.
No
Please.
problem. Yeah, please, Nilanjan.
Pratyush, always great to hear you. A quick question. So, of course, as you explained that when we are looking at reaction coordinates and we are looking at, the maxima, if you will, they may not necessarily be transition states. That's well taken, of course. And we may not be looking at the committers, right? So I was wondering if you guys also have some methods to actually track down, the transition state as realistically as possible. Because sometimes that is important. We are mostly
No,
focused
so
on.
you get it as a free byproduct of the state predictive information bottleneck. I don't think I showed that in one or let me
Yeah, I
show
would
you.
appreciate, a little bit
No, no,
more.
sure. So that's this. I'm looking for my. I had thought I will not show alanine dipeptide, but since you asked, now I will have to show alanine dipeptide. So, and nothing against
You
alanine
wrote a
dipeptide.
very awesome paper, which I really make some of my students read, is when you have phi and psi and you sample for the phi, and then how can you get the psi back? So that was pretty
Yeah,
cool.
that's no, but there's in the information bottleneck approach. So I think this is related to what you're asking, Lanjana. So when we start the simulation, we don't know how many states are there. So we just say, okay, let's there are 10 states, right? And you can do this along phi. You can do this along psi, the initial state demarcation. Now, when you predict the information bottleneck, when you use the information bottleneck to predict this system 0.5 picosecond into the future, the population out of these 10 states, 7 of them disappears and you're left with 3. And this boundary is actually the committer, 50% committer boundary between these two states. So this boundary, so our method gives you the 50% committer boundary, which is the transition state by construction, while this boundary is the 50% committer boundary between phi and psi. And this is and you can actually do the committer calculation and show that it is as sharply peaked as it's going to be given these things. So you are getting the transition state out of this. I think I have this one also where I can compare. I mean, the most accurate method for getting transition state is likelihood maximization from Baron Peters and others. And here I'm showing you that for this ion pair dissociation problem, the transition state actually does not involve just the distance between the two ions. It also involves the number of bridging waters between these. And you can see that the transition state coming from likelihood maximization shows you that there is a peculiar distribution of the transition state ensemble where you have a dependence between R ion and the number of bridging waters, and we get the same distribution. So you get this out of information bottleneck.
Great. Thank you, Pratyush.
Well, whether it's the fastest method to do that or not, that's another story, but you do get it out of it.
Okay. Yeah. Anything more, Nilanjana? You may want to say hi to a few other people like Suman and all. I can see them in the audience.
I can see them hanging in the background.
Yeah. If there are, yeah, he says hi. He's on the road. It looks like he's traveling, so he may be connected through phone. Any other questions from students, postdocs? Please feel free. I've also put in Pratyush's email ID. If you want to send him an email about tools or questions, I'm sure Pratyush is more than happy.
Yeah, I will be more than happy to.
Pratyush is one of those guys who believe in democratizing science and has a very altruistic attitude towards science, which is something very inspiring for all of us.
No, don't be so kind.
That's true, Pratyush. I mean, it's a community, right? So it's great to have. As Jagannath apologized, he had to leave because
he's traveling.
Okay, if there are no more questions, then I think we'll thank Pratyush for a wonderful talk and a very uplifting talk. That and a lot of things to you mull about and try to think how we can get into our own work. So thanks a lot, Pratyush. Hopefully,
thank you, Anand. Thank you, Jagannath.
Around. Yeah.
Okay. Bye. Have a nice evening.
Thanks. Have a great day. Bye, everybody, for joining us. Appreciate you coming and engaging with us. Thanks. Bye.